Welcome to our comprehensive guide on gene annotation techniques. In this video, we'll break down what gene annotation is, why it's crucial in genomics, and how scientists use various methods to interpret genetic information. Whether you're a student, researcher, or just curious about genetics, this journey will help you understand the basics and the latest advancements in gene annotation. Let's start by exploring the fundamental concept of gene annotation and how it fits into the broader field of genomics. Gene annotation is the process of identifying and marking the locations and functions of genes within a genome. It's like adding notes to a complex instruction manual, helping researchers understand which parts of DNA code for proteins, regulatory elements, or other important features. This process is essential for making sense of raw genetic sequences and turning them into meaningful biological information. Gene annotation is vital because it bridges the gap between raw DNA sequences and biological function. Without annotation, a genome is just a long string of letters. Annotation reveals which segments are genes, what they do, and how they interact. This knowledge is crucial for medical research, agriculture, and understanding evolution. Accurate annotation can lead to discoveries about disease mechanisms, new treatments, and improved crops. There are two main types of gene annotation: structural and functional. Structural annotation focuses on identifying gene locations, exon-intron boundaries, and regulatory elements. Functional annotation, on the other hand, assigns biological meaning to these features, such as predicting protein function or involvement in pathways. Both types are essential for a complete understanding of the genome. Structural annotation is the process of pinpointing where genes and other elements are located within the genome. This involves identifying start and stop codons, exon and intron regions, and regulatory sequences. Scientists use computational tools to scan DNA sequences and predict these features, often comparing them to known gene models from other organisms to improve accuracy. Functional annotation goes beyond structure, aiming to describe what each gene does. This includes predicting the function of proteins encoded by genes, their roles in biological pathways, and their interactions with other molecules. Functional annotation often relies on databases of known proteins and experimental data to make accurate predictions. Manual annotation involves experts reviewing genetic data and making informed decisions about gene locations and functions. This method is time-consuming but highly accurate, as it allows for the integration of experimental evidence and expert knowledge. Manual annotation is often used to validate computational predictions or to annotate genes in newly sequenced organisms. Automated annotation uses computer algorithms to analyze large volumes of genetic data quickly. These tools can scan entire genomes, predict gene structures, and assign functions based on similarity to known genes. While faster than manual annotation, automated methods may miss subtle features or make errors, so results often require further validation. Gene prediction algorithms are at the heart of automated annotation. These programs use statistical models and machine learning to identify likely gene regions within DNA sequences. By comparing patterns in the sequence to known gene features, they can predict where genes start and end, as well as their internal structure. Homology-based annotation relies on comparing new genetic sequences to those already annotated in other organisms. If a gene in one species is similar to a known gene in another, it's likely to have a similar function. This method is powerful for annotating genes in newly sequenced genomes, especially when closely related reference genomes are available. Ab initio annotation predicts genes based solely on the DNA sequence itself without relying on external data. Algorithms look for patterns like open reading frames, start and stop codons, and splice sites. While this method can identify novel genes, it may also produce false positives, so results are often combined with other techniques. RNA sequencing (RNA-seq) is a powerful experimental technique for gene annotation. By sequencing the RNA produced in a cell, scientists can identify which genes are actively expressed and map their exact boundaries. RNA-seq data helps refine gene models and discover new transcripts, making it an essential tool in modern annotation pipelines. Proteomics, the study of proteins, provides direct evidence for gene annotation. By identifying proteins present in a cell, researchers can confirm which genes are actually translated and refine their annotations. Protein evidence is especially valuable for validating predicted genes and understanding their functional roles. Gene Ontology (GO) annotation assigns standardized terms to genes, describing their biological processes, cellular components, and molecular functions. GO annotation helps organize genetic information and makes it easier to compare genes across different species and studies. It's a key part of functional annotation workflows. Pathway annotation links genes to specific biological pathways such as metabolism or signal transduction. By mapping genes to pathways, researchers can better understand how genes work together to carry out complex functions. This approach is especially useful for studying diseases and identifying potential drug targets. Several public databases store gene annotation data, including GenBank, Ensembl, and RefSeq. These resources provide access to annotated genomes, gene models, and functional information. Researchers rely on these databases for reference, comparison, and further analysis of genetic data. Community annotation projects involve researchers from around the world working together to annotate genomes. By pooling expertise and resources, these projects can produce high-quality annotations and keep data up to date. Examples include the Human Genome Project and various model organism databases. Ensuring the accuracy of gene annotation is critical. Quality control measures include manual review, cross-validation with experimental data, and comparison to known gene models. These steps help minimize errors and ensure that annotations are reliable for downstream research. Gene annotation is not without challenges. Complex genomes, repetitive sequences, and limited experimental data can make accurate annotation difficult. Additionally, differences between species and ongoing discoveries mean that annotations must be regularly updated and refined. Not all important DNA sequences code for proteins. Non-coding regions such as regulatory elements and non-coding RNAs play vital roles in gene expression and genome function. Annotating these regions is challenging but essential for a complete understanding of the genome. Comparative genomics involves analyzing multiple genomes to identify conserved genes and features. By comparing genomes from different species, scientists can improve annotation accuracy and discover evolutionary relationships. This approach is especially useful for annotating poorly characterized genomes. Alternative splicing allows a single gene to produce multiple transcripts and proteins. Annotating these variants is important for understanding gene function and regulation. RNA-seq data is particularly useful for identifying and annotating alternative splicing events. Small RNAs, such as microRNAs and cRNAs, play key roles in gene regulation. Annotating these elements requires specialized techniques and experimental data. Understanding small RNA annotation helps reveal new layers of gene control and cellular function. Epigenetic annotation involves mapping chemical modifications to DNA and histones that affect gene expression without changing the sequence. Techniques like ChIP-seq are used to identify these modifications, providing insights into gene regulation and cellular identity. Model organisms like mice, fruit flies, and yeast have well-annotated genomes. These species serve as references for annotating genes in other organisms. Model organism databases are valuable resources for comparative annotation and functional studies. Plant genomes present unique challenges for annotation due to their size, complexity, and repetitive elements. Specialized tools and databases have been developed to annotate plant genes, supporting research in agriculture and crop improvement. Microbial genomes are generally smaller and less complex, making annotation more straightforward. However, the vast diversity of microbes means that many genes have unknown functions. Ongoing annotation efforts are essential for understanding microbial roles in health and the environment. Human genome annotation is a major focus of biomedical research. Accurate annotation helps identify disease genes, understand genetic variation, and develop personalized medicine. Projects like ENCODE and GENCODE provide detailed human gene annotations for researchers worldwide. A variety of software tools are available for gene annotation, ranging from command-line programs to user-friendly web interfaces. Popular tools include AUGUSTUS, Maker, and GeneMark. These tools automate many aspects of annotation and are continually updated to incorporate new methods. Visualization tools help researchers explore and interpret gene annotations. Genome browsers like UCSC and Ensembl allow users to view annotated genomes, compare features, and integrate experimental data. Effective visualization is key for making sense of complex annotation data. Modern annotation pipelines often integrate data from multiple sources, including genomics, transcriptomics, proteomics, and epigenomics. Combining these data types provides a more complete and accurate picture of gene function and regulation. Machine learning is increasingly used in gene annotation to improve prediction accuracy. Algorithms can learn from large datasets, identify complex patterns, and adapt to new data. This approach is helping to automate and refine annotation processes. Crowdsourcing involves engaging the broader scientific community or even the public in annotation tasks. Platforms like GeneWiki allow users to contribute knowledge and improve gene annotations collaboratively. This approach leverages collective intelligence for better results. Personalized medicine relies on accurate gene annotation to interpret individual genetic differences. By linking genetic variants to annotated genes and functions, doctors can tailor treatments to each patient's unique genetic makeup, improving outcomes and reducing side effects. Gene annotation raises important ethical questions, especially regarding privacy and data sharing. Researchers must ensure that genetic data is handled responsibly and that individuals' rights are protected. Ethical guidelines and regulations help guide annotation projects. The future of gene annotation is bright, with advances in sequencing technology, artificial intelligence, and data integration driving progress. As new methods emerge, annotations will become more accurate, comprehensive, and accessible, opening new doors for research and medicine. Training the next generation of scientists in gene annotation is essential for continued progress. Educational programs, workshops, and online resources provide hands-on experience with annotation tools and techniques, preparing students for careers in genomics. Open science initiatives promote the sharing of annotation data and tools, making research more transparent and collaborative. Publicly available resources accelerate discovery and ensure that everyone can benefit from advances in gene annotation. Accurate gene annotation is particularly important for studying rare diseases. By identifying disease-causing genes and variants, researchers can develop better diagnostics and treatments, offering hope to patients and families affected by rare conditions. Cancer genomics relies on detailed gene annotation to identify mutations, understand tumor biology, and develop targeted therapies. Annotated cancer genomes help researchers pinpoint driver mutations and design personalized treatment strategies. Gene annotation plays a key role in understanding infectious diseases. By annotating pathogen genomes, scientists can identify virulence factors, drug resistance genes, and potential vaccine targets, aiding in the fight against global health threats. Gene annotation is fundamental for evolutionary biology. By comparing annotated genomes across species, researchers can trace the origins of genes, study evolutionary changes, and uncover the genetic basis of adaptation and diversity. Synthetic biology uses gene annotation to design and build new biological systems. Accurate annotation ensures that synthetic constructs function as intended and helps scientists engineer organisms for applications in medicine, industry, and the environment. Gene annotation is a dynamic field with new discoveries and technologies constantly emerging. Regular updates to annotation databases and tools are essential to keep pace with scientific progress and ensure that researchers have access to the latest information. In summary, gene annotation is a cornerstone of modern genomics, enabling scientists to interpret genetic information and drive discoveries in health, agriculture, and beyond. As techniques continue to evolve, the accuracy and utility of gene annotations will only increase, paving the way for new breakthroughs and a deeper understanding of life itself.